Good morning, viewers. Today we are going to get into the topic DNA vectors. We are going to understand the basics involved in the DNA vector. Let's enter into the topic. Coming to the introduction of DNA vectors, what are DNA vectors? DNA vectors are self-replicating DNA molecules that carry foreign DNA into a host cell. The DNA vectors are used to clone, amplify, and express a gene of interest and helps in successful propagation of the vector into another host system. We'll have a question, why are these vectors important? They are important in various fields like genetic engineering, biotechnology, medicinal research, and agricultural applications for generation of new varieties of crops, medicines, and other useful products. Let's see what are all the key components of a DNA vector. The DNA vectors has multiple key components like origin of replication, selectable marker, multiple cloning site, promoter, and terminator. These components helps the DNA vector for successful sustainment in the host cell and transformation to the next generation. Origin of replication, also called as ORIC, enables autonomous replication within the host cell. The selectable marker, which is a gene which confers resistance to antibiotics or other selective agents, which is used to identify cells containing the vector. The multiple cloning site, MCS, is a region that contains multiple restriction enzymes recognition site, which allows insertion of a foreign DNA fragment. The promoter is a DNA sequence that initiates transcription of a gene and which is required for gene expression. And the terminator is a DNA sequence that signals the end of a transcription, which helps in protein synthesis and processing in a host cell. Let's see about the different types of DNA vector. Plasmids. So plasmids are circular double-stranded DNA molecule which are commonly used in bacterial cell. Examples of artificial plasmids include PBR322, PUC19. And next category of DNA vectors are bacteriophages. So these bacteriophages are nothing but the virus that can infect the bacteria, and they are highly efficient in the process of infection and replication. Examples of DNA vectors of bacteriophages are lambda-phage and M13 phage. Cosmids. Cosmids are hybrid vectors where the DNA is shuttled between a plasmid and a phage molecule. They can carry larger DNA inserts. Yeast artificial chromosomes, also called as YACs, can accommodate very large DNA fragments which are used for cloning complex genomes. Bacterial artificial chromosomes, also called as BACs, can carry larger DNA inserts. They'll be stable due to the presence of the centromere and are easy to manipulate. These are all the different types of DNA vectors. Let's get into the applications of the DNA vectors. The various applications of DNA vectors include gene therapy, protein production, vaccine development, agricultural biotechnology, and forensic sciences. So these DNA vectors are used in the field of cloning for synthesis of various valuable products in different fields such as gene therapy and protein production. So in conclusion, DNA vectors are essential tools in modern biotechnology. They enable the manipulation and study of genes. Their applications are diverse and far-reaching. Thank you for your patient listening. Let's get back with another interesting topic soon.